In osmosis, water is always flowing in the direction of from an area of high concentration to an area of low concentration. Because of this, the type of the solution that the cell is in can greatly affect how and where the water is going to flow due to osmotic pressure. So we're going to talk about some different types of solutions. One solution could be a hypertonic solution. Hyper meaning greater than, and tonic referring to the strength of the solution. Hypertonic means that the solution has a higher concentration of solutes on the outside than on the inside of the cell. In this case, we're going to be referencing a human cell. So if you were to take a human cell, you were to put it into a cup of highly concentrated salt water, what is going to happen to the cell? What we'll find is that water is going to rush out of the cell in order to kind of equilibrate. We want the concentration on the inside of the cell to be the same as the concentration on the outside of the cell, so we're going to try and reach equilibrium by water rushing out of the cell. This is going to cause the cell to shrivel up. Any solution with a concentration greater than 0.1 sodium chloride is going to be hypertonic to a red blood cell. Animal cells placed in such a solution are going to shrivel up due to water loss. This process is referred to as crenation in red blood cells. In a plant cell that's placed in a hypertonic solution is going to experience plasmolysis. This is where the cytoplasm is going to shrink away from the cell wall, and it's going to actually pull away, and the plant is going to start to wilt. So that's a hypertonic solution. Another type of solution would be an isotonic solution. In an isotonic solution, iso meaning same, tonic referring to that strength. In this case, an isotonic solution has the same concentration of solutes on the inside as the outside of the cell. In this case, there's going to be no net movement of water. Because the concentrations are the same, water is going to move in at the same rate that it's moving out. Does anybody know or can give me an example of an isotonic solution that we use when dealing with animal cells? What is another name for the isotonic solution that we use most frequently? And I'll give you a hint. It's a solution that is most frequently used in IVs. Because what would happen if we were to inject pure salt water into our veins? If we were to go get some ocean water and inject that into our veins, what would happen to our cells? And conversely, what would happen if we injected pure water into our veins? We don't. We tend to inject an isotonic solution. So brownie points for anybody who can give me the name of that solution that we use. A hypotonic solution, hypo meaning less than, tonic referring to that strength. This is going to have a lower concentration of solutes on the outside than the inside of the cell. In an isotonic solution, the solute concentration and the water concentration are going to be completely equal. So The solute concentration is going to be lower on the outside of the cell than the inside of the cell. As we mentioned before, the solute concentration of red blood cells is 0.1%. Thus, any solution that has less than a 0.1% exposed to human cells is going to result in something known as cytolysis, which is the splitting open of cells. In a hypotonic solution, those animal cells might even burst. Cytolysis, cyte meaning cell, it's the same root word as the word cytoplasm, and lysis, to burst, to break apart, to split. In red blood cells, this is what's known as hemolysis, heme referencing the blood. Red blood cells placed in a hypotonic solution are actually going to take on so much water that they swell up and burst. In plant cells placed into a hypotonic solution are also going to swell. However, this isn't a dangerous situation for plants. You can take fresh cut flowers and put them into a vase of fresh water. And this is actually a good thing, because due to the cell wall found on the outside of plant cells, the cell is not actually going to burst. It will swell up, it will fill up, but this is actually very important. It's called turgor pressure. It allows the plant to maintain its posture and maintain its structure and keeps the plant rigid and upright. So not all hypotonic solutions are bad. It just depends if you're a plant or an animal cell and if you have that rigid cell wall to protect your cells from bursting. Oh, hypotonic solution water is going to move into the cell. So here's an example of what all of that looks like. Here we have a bag of water that is 98% sugar and 2%, or 98% water, 2% sugar. This bag is going to represent a semi-permeable membrane. It's going to allow water to move in and out, but not sugar molecules. So what happens when we place this bag in a hypertonic solution, more water is going to rush out than rushes in. The bag is actually going to shrink up as it tries to reach equilibrium with the solution around it. This is what a red blood cell looks like when it has been placed in a hypertonic solution. You can see that these cells have started to shrivel up. They don't have that nice round donut shape anymore. In an isotonic solution, we're going to see an equal amount of water going in as going out, and in and out. We're in perfect equilibrium. So an isotonic solution is going to have the same concentration, as the cell in our model. In the case of a hypotonic solution, if we were to put this bag into 100% distilled water, we're going to see the cells actually swell up. And if they swell up far enough, they may actually burst. So you can see it's a little bit less noticeable than the cells that have undergone crenation, but you can see that these donut well dips are not as severe or as pronounced as they are over here. So these cells have swelled up and started to round a little bit. And that's going to be it for our passive transport. And the next thing we want to discuss is active transport. There are times when the cell may need to increase its concentration gradient across the membrane in order to accomplish more work. So in order to do this, we're actually going to have to move molecules against the concentration gradient. This is what's known as active transport. Moving against the concentration gradient is kind of like moving against the current. You can sit in the current and you can float. If you don't put in any effort, you are going to float with the current of a river. You're going to flow downstream. If, however, you wish to go upstream, you're going to have to expend a little bit of energy. You're moving from an area or you're moving up against the current. In the case of active transport, we're moving from areas of low concentration to high concentration. This is almost always going to require the aid of a carrier protein and energy. Typically, the energy that we're going to be using is ATP. This is going to help bind to substrates and help move them across the plasma membrane. Here we have an example of what that looks like. We have a low concentration over here, a high concentration of molecules here, and what we're going to need to do is bind to the molecule over here on this side of low concentration. And once it's bound in, we're going to need ATP to undergo a conformational change, allow this carrier protein to snap shut on this side, open up on this side, and release its cargo. In order to do this, we're going to need that ATP. We're going to need that energy in order to pick up and move these molecules against their concentration gradient. But there are other forms of active transport, and these are, of course, going to require ATP as well. But they are utilized for moving molecules that are much larger than can be handled by a carrier protein. These are what are known as macromolecules. Macromolecules like proteins, polysaccharides, nucleic acids, they're going to be way too large to be transported into or out of the cell by just a carrier protein. They're going to require bulk transport. Bulk transport moves in two directions: out of the cell in exocytosis, this is macromolecules exiting the cell, and endocytosis, macromolecules entering the cell. So we can move in both directions. We can either secrete proteins or we can pick them up. We can engulf bacteriophages, or we can break them down, or we can release large molecules. So we can move into and out of the cell in bulk via exocytosis and endocytosis. We're going to start off by talking about endocytosis. In endocytosis, a region of the plasma membrane is going to reach out, engulf the substance to be ingested, and then pinch off from the rest of the membrane to enclose that substance in a vesicle. That vesicle is then going to travel through the cytoplasm to wherever it is that it needs to go. This process, although we don't see it directly, is all orchestrated by the cytoskeleton. The cytoskeleton is going to need huge amounts of ATP to actually carry this out. So although you may not see in our models a molecule of ATP being expended, just note that this whole thing is orchestrated by the cytoskeleton. Just like it requires energy to move your body, to move your skeleton, it's going to require energy to move the cell skeleton or the cytoskeleton. There are two types of endocytosis, depending on what it is that we want to accomplish. There's phagocytosis, also known as cellular eating. This is when the cell is ingesting large particles or bacterium. Pinocytosis is similar, but in this case, this is cellular drinking. We're picking up droplets of fluid that have small substances dissolved into them. So phagocytosis is like taking a bite of a sandwich. Pinocytosis is like taking a gulp of water. And here's just what that looks like. Here we're going to see the plasma membrane is actually going to have to reach out, reach around, engulf a bacterium, bring it inside, and pinch off that plasma membrane. Once we have this vesicle brought inside the cytoplasm, it can be transported to wherever it needs to go into the cell, or hydrolytic enzymes can be allowed to pass through this plasma membrane and start chomping down, breaking down that bacteria. Same with pinocytosis. We're going to reach out, we're going to engulf an amount of that extracellular fluid, and we're going to bring it inside in one of our little vesicles. Exocytosis is going to look very similar, but it's going to be just in the opposite direction. Exocytosis is when we are sending large molecules out. They're going to be enclosed in a membrane-bound vesicle. That vesicle is going to travel up to the plasma membrane, where it will then be released to the outside. Here's an example of that. Here is our vesicle carrying our precious cargo. And what sort of cargo might we want to send out of the cell? We can allow certain amounts of waste to pass through our cell membranes. Specifically, carbon dioxide can diffuse through that plasma membrane very easily. But larger molecules, larger wastes, are going to have to be brought to the surface in a vesicle. This vesicle may also contain things like proteins to be secreted. Human insulin is a protein. It's used to help regulate blood sugar levels. It's going to be produced in cells, specifically in it's going to be produced in cells in the pancreas, and it's going to then be secreted out of those cells into the bloodstream and travel to where it is that it needs to go. And so this is one way that we can get those proteins, those large molecules out of the cell, is by bringing them up to the surface. This membrane that surrounds the cell can fuse directly with the plasma membrane to release its cargo on the other side. Here is a great study table, a great summary of everything that we have talked about. I would spend a little bit of time reviewing table 3.2. It's going to break down simple diffusion, facilitated diffusion, osmosis, active transport, endocytosis, and exocytosis in a very concise way. This might be a great place to get definitions for study guides or study aids if you like to make flashcards or whatever form you like to make your study tools. So spend a little bit of time reviewing table 3.2 before you move on to our next sections. All right, next up we're going to be studying our organelles. So take a moment, spend a little time reviewing osmosis. If you have any questions about any form of active or passive transport, please feel free to shoot me an email at any time, and I will do my best to either answer in an email or answer in one of our study discussions. I'm always happy to make more videos to help explain things further. All right.